Okay, good morning. Thank you for coming, and thank you to the organizers for inviting me. The title of my talk is Mycorrhizas and Changing Ecosystems, which refers directly back to a paper in 1991 by David Reed titled Mycorrhizas and Ecosystems. And I refer to that paper because it was a really important paper in our thinking, and I think it's still very relevant. What David said was that in nature, each mycorrhizal type is associated with an ecosystem and soil environment with distinctive characteristics. And he actually went so far as to draw this remarkable map of the world in which he divided the world up by mycorrhizal type. Literally a black and white view of ecosystem function. And I have to say, I took that view very much on board. Where previously I had seen systems as comprising of plants, grasses, and trees, I actually began to see the world in a black and white sense as being ectomycorrhizal or arbuscular mycorrhizal, and the two as very different things that didn't function in the same way. And yet, ecosystems change. This is a grassland in New Zealand. It's arbuscular mycorrhizal. It's being invaded by Pinus contorta, which is shifting it into an ectomycorrhizal system. That same spot, if I could take you back in time 300 years, you would have found was dominated by ectomycorrhizal trees prior to European clearing. And if we were to go back even further through time, looking at pollen core data, we would find that it had been dominated by an arbuscular mycorrhizal forest of podocarps and only became ectomycorrhizal maybe 7,000 years ago. And so the system may have once looked like this. And it's shifted through time from arbuscular mycorrhizal to ectomycorrhizal to an arbuscular mycorrhizal grassland. It is now returning to being ectomycorrhizal. And once you start looking for these ecosystem shifts, you see them everywhere. You see them in clearing of land for agriculture, and conversely, planting of ectomycorrhizal trees into grasslands, like here in Patagonia. You also see them in many invasive species. This is an Araucaria forest being replaced by invasive ectomycorrhizal pines, and equally with pathogens, which can remove different mycorrhizal types from an ecosystem, like sudden oak death or a number of others. And I would argue that in this era of global climate change and globalization, we are actually likely to see an increase in the rate of ecosystem change. Now, if you take the view that ectomycorrhizal systems are fundamentally different from arbuscular mycorrhizal systems, then these changes, these shifts in mycorrhizal type, should have profound effects on ecosystem function. And yet when you go to ecologists and you try to say, how will this change the ecosystem, they can't tell you. And the reason is because ecologists continue to ignore mycorrhizas. If you look at all of the major nutrient cycling models that are used to predict ecosystem function, none of them really include mycorrhizal status. At best, they include mycorrhizas as one thing. And so the question for me becomes then, what happens when an ecosystem shifts in mycorrhizal status, which is really fundamentally asking, what does it mean to be ectomycorrhizal? Or conversely, what does it mean to be arbuscular mycorrhizal? And by asking that question, I'm treading very close to the dangerous question, do mycorrhizae even matter at all? So there are two things that really stand out that people have mentioned about ectomycorrhizal systems. One is that they often have a quite high level of organic matter in the soil and an accumulation of litter. And the other that's quite marked is that they're often quite low plant diversity. So as little as a single tree species dominating the forest. That's true in the temperate zone, like in this Kunzea stand in New Zealand, but it's perhaps nowhere more apparent than in the tropics, where you get ectomycorrhizal monodominant stands surrounded by diverse arbuscular mycorrhizal forests. Of course, there are exceptions, like the dipterocarp forest of Southeast Asia, or tundra vegetation, which has an amazing diversity of species, but even there, all of the species tend to be ectomycorrhizal, or ericoid mycorrhizal. And so, although it's not low diversity, it's at least low diversity in the sense of having a very high dominance of ectomycorrhizal plants. So what explains these patterns? Well, there have been four major hypotheses proposed, and I'm going to go through those in my talk one by one. The first is looking at whether ectomycorrhizal plants have distinctive foliar characteristics. Then I'll move from that to look at plant-soil feedbacks and mycorrhizal networks. Having covered those two, I'll move on to looking at interactions with minerals and rock-eating fungi. And finally, look at the role of ectomycorrhizal fungi in organic nutrient cycling. So starting looking at foliar characteristics, this hypothesis actually goes back to David Reed's 1991 paper, but he phrased it nicely in 2004, where he said that plants with ectomycorrhizae have lower nutrient concentrations and growth rates than our mycorrhizal plants, and also very recalcitrant litter. In 2001, Hans Cornelissen actually tested this hypothesis, looking at the flora of Britain and looking at foliar characteristics like plant growth rate, foliar nitrogen, foliar phosphorus, and decomposition rates. And he was able to show that mycorrhizal status was a significant predictor of some of these traits. And so perhaps we should just take this hypothesis and give it a big red check and say, yes, that's confirmed. But it might be a bit premature to do that. If we look at the data that Cornelissen used, he restricted himself to the British Islands, and he actually only had 12 ectomycorrhizal plants, of which 7 were in the Phagales and 3 in the Pinaceae. And I'm not sure that we can really extrapolate from a data set of 12 species to the 6,000 or more species of ectomycorrhizal plants around the globe. Now, since 2001, there's been an explosion of research on plant foliar traits, particularly led by Peter Reich, Mark Westaby, and Ian Wright. And they've been looking at traits like foliar nitrogen and phosphorus and trying to look for predictors of them. And they've been quite successful at showing that a number of plant traits are strong predictors of foliar nutrients. And yet they had never looked at mycorrhizal status in any of their analyses. So we wanted to return to the Cornelissen type of question, but take it up to the level of the globe. So we worked with Peter Reich, took his entire data set. We ended up with 11,000 observations representing more than 3,000 species, so a fairly significant data set. And we simply asked, do ectomycorrhizal plants differ in their foliar traits from our mycorrhizal plants? If you just ask that question of the raw data, and throw all the... But we found very significant responses for phosphorus, the nitrogen-phosphorus ratio, and leaf mass per unit area, which is a measurement of leaf thickness. But there's a big issue in this sort of analysis, which is that it's a very biased data set. In fact, it's largely been collected by foresters, and so it's dominated by data from pine trees. So what we did to address that was we took a phylogenetic approach where we went through the entire plant kingdom and we identified clades of ectomycorrhizal plants. In total, we took a very conservative approach tending to lump things together. We identified 19 ectomycorrhizal clades, and then we could do a pairwise comparison of the plant traits of each one of those clades and compare it to the ancestral state that was either arbuscular or non-mycorrhizal. And we could plot these in a one-to-one plot, and with the ectomycorrhizal trait on the x-axis, on the y-axis, and the non-ectomycorrhizal trait on the y-axis. If there was no difference, you would expect the trait values to fall on a one-to-one line. If the ectomycorrhizal trait was higher, you'd expect it to fall above the line, and lower, you'd expect it to fall below the line. I'll just show the significance by shading. So our hypothesis was that ectomycorrhizal plants would fall on one side of the line or the other. And here's what we found. Pretty much a cloud of data points with no pattern at all. In fact, a number of species were significantly above the line, but there were also quite a few significantly below the line. And that was true for nitrogen, phosphorus, the ratio between the two, and leaf mass per unit area. You can test that statistically using a t-test or a sign test. We did both, and we found not one of the traits showed any significant pattern. The peer reviewers asked us to come back with some more tests, so we tested that looking only at groups where we had reasonable amounts of data. That was an n value of greater than 10. We still had no significant pattern. The peer reviewers asked us to come back with more statistics, so we did a null model with randomization of the data and also a true phylogenetic independent contrast, which takes the entire phylogeny into account, and we still found no significant pattern. That's less significance than you would expect by random chance. So I'm fairly confident that we can say there is no evidence for any consistent patterns in foliar traits with mycorrhizal status. So I'm going to suggest that we reject that hypothesis. The second hypothesis is looking at plant soil feedbacks. Now this goes back to some work by Jim Beaver in the late 90s, where he developed these very complicated statistical models of feedback. They're very hard to understand, honestly, but they're very easy to do, which is that you take field soil, you culture it with a plant species, and then you plant either the same species or a different species and look at the relative fitness of those seedlings. And he's done some superb work in arbuscular mycorrhizal fungi, showing that plants tend to accumulate fungal communities that are actually more beneficial to other plant species than to their own seedlings. More recently, that's been applied to trees by Scott Mangan in a neat paper looking at tropical rainforests, where he's shown that all 5 of the species he looked at, these were tropical rainforest trees, showed negative feedback, and that was strongly correlated with the abundance of the trees. This is important because it ties into something called the Jansen-Connell hypothesis, which can be paraphrased as saying that negative feedback is an important force maintaining tropical forest diversity. Now, in my own work, it's very difficult to grow trees in pots for 2 generations within a reasonable research project. So what I've tended to do instead is to look at distance transects from forest edges moving out into grasslands as a measurement of tree-seedling interactions. And I'll just show one little piece of data. It's from a study I did in Minnesota, where we looked at mycorrhizal infection of bur oak seedlings as a function of distance from the forest edge in 2 different fields, left and right, and in 2 different years. And what we found consistently was that mycorrhizal infection was high near trees, stayed high for some distance, and then dropped off quite dramatically. And that was correlated with growth as well. And so I took this as evidence of positive feedback, the opposite pattern as described in these other systems. Now, Jim had taken his data and built a spatial model of what the effects of positive and negative feedback might be on plant communities. It's a strange model because it's spatial in 1 dimension, left to right like a slice through a plant community, and then you track through time as you go up. And what he showed is that negative feedback, you've got plant communities moving around and having fuzzy edges, so fairly high diversity, but positive feedback led to the maintenance of patchy ecosystems with very low diversity within them, which led me to hypothesize that positive feedback between ectomycorrhizal trees and seedlings might contribute to increasing the importance of ectomycorrhizal trees in ecosystems, or in other words, might contribute to ectomycorrhizal monodominance. But there was something that bothered me, which is that my data showed a strange pattern. It was that when you were near a tree, it was good, but being closer to the tree or near 2 trees was not any better. Whereas the spatial models that I was basing my hypothesis on assumed that if you were near 1 tree, it was good. If you were near 2 trees, it was twice as good. If you were near 3 trees, it was 3 times as good. So I wanted to look at how that assumption might influence the outcome. So I built a model. It's a simple spatial model. I put 5 species into it, represented by different colors, killed off 5% of them every year, and then I allowed a feedback to occur where the neighbors influenced the probability of regeneration, and ran that for 2,000 cycles, each cycle killing off seedling or spots and then replacing them. For my response variable, I look at the number of species in every 9 cells square. So it's sort of measurement of local diversity. That's what it looks like when you throw it up on the computer screen. It's quite a pretty pattern. Then you can run it, and you see the species shifting around. You spend hours sitting there meditating, looking at these things running. But what you see in terms of the results is that diversity starts out pretty high, and it pretty much stays high. You get a maintenance of diversity without any feedback. What if you take the same model and run it with a positive feedback? So here I'm allowing the neighbors have an additive positive effect, just like in Jim Beaver's model. And what you see very quickly is that you develop these monodominant patches, and you get a loss of diversity. And looking at the response metric here, you see it drops very quickly down to 1, or very close to 1. And that's showing exactly what Jim already showed in his model, except mine's prettier. So then I wanted to look at what happened if you put in a non-additive function, so the positive feedback that didn't add up, so that being near 2 was just as good as being near 1. And unfortunately for me, it didn't change anything. In fact, it was just like positive feedback. So whether it was additive or not additive made very little difference. It was slightly slower, but seemed to not matter. But there's another problem with my data, which is that the positive feedback that I saw extended a considerable distance from trees out into the grassland. And that makes sense. If you look at trees, they have roots that are often much longer than the tree is tall. So here I am holding a pine tree in my right hand. My arms are stretched out because that's one root that I'm stretching out from it. Here I am with a washed up willow tree from a river valley in New Zealand. And again, the roots extend far beyond the tree. So I wondered what would happen if you took this model and you ran it allowing the positive feedback to extend not just 1 cell, but 3 cells. So a longer distance positive feedback. And what you see when you run that is that you don't get nearly as much patchiness developing. And in fact, the non-additive function over long distance does not lead to a loss of diversity. And so on the basis of the model combined with the field data, I would have to say that increased mycorrhizal inoculum is not likely to lead to monodominance, and that's for two reasons. One is because it's a non-additive function, but also because it's a long-distance function. And so I have to take the hypothesis of Dickey et al. 2002 and reject it. I get to reject my own work. But before we move on from feedback, I want to look at a recent paper that came out in Science, where they also looked at feedback in forest systems. What Johnson et al. Did was they took 200,000 forest plots from the eastern North America, and they simply correlated the abundance of seedlings of a species with the abundance of trees of that species, okay? For every one of those spatial grid cells. And what they found was remarkable. They showed an overwhelmingly negative feedback, which means that the slope of those relationships was almost always negative. It was highly significant, and importantly for me, it left no scope for any positive effects whatsoever. There was virtually no data above zero, which meant that everything that I had done must have been wrong. This is critical because half of their data, slightly more than half of their data, were ectomycorrhizal species. So they concluded on the basis that negative density dependence or negative feedback was a pervasive mechanism driving forest diversity. But this result was remarkable because it actually was so strong that what they were saying was that negative feedback overwhelmed gravity. It overwhelmed local seed dispersal. And it also didn't confirm with my own idea that in forests you often see seedlings of a species under trees of the species, not vice versa, which is what their result would predict. So I decided to test that a little further. Their data represented mostly young forest plots, so I thought I would test it in a data set from New Zealand where we had over 1,100 forest plots, so a smaller data set, but where many of our forest plots were over 100 years old. And when I tested it using the same feedback mechanism as they did, I found actually a slightly positive feedback, not big, but it was significantly positive. So I wrote an entire paper based on that result, and I went to bed, and I lay there and I thought about my data. And this is made up data here, but it's just to prove a point, or to show a point, which is that this is the way the data were recorded in both data sets. What you saw was that for Pinus strobus in plot 1, you had 0 seedlings, 3 trees. In plot 2, you had 3 seedlings, 2 trees. Plot 4, you had 2 seedlings, 0 trees. You notice that plot 3 is missing. If a species wasn't present as either a seedling or a tree, it was not in the data set. That plot was not put in. So that seedlings could only have a 0 value when the tree value was greater than 0. And if you think about the effect of that, you might look at data and see what you think is a negative feedback. But if you were to add in a whole bunch of data points at the origin, it might reverse that feedback. And so I reanalyzed the data, inserting in the joint abscesses, putting back the data that had been left out. I generated an entirely random data set, and here's what I got. Very strong negative feedback following the same exact pattern as had been reported in the forest data. When I analyzed that correctly, including the 0-0 data, of course you saw no pattern. It was random data. You expect no pattern. In the New Zealand data, where you remember I saw a slightly positive feedback of excluding the joint absences, when I reanalyzed it, including joint absences, it was 4 times more positive. And in the North American forest data, where they had reported a slightly negative feedback, but highly significant, when analyzed including joint absences, you get a complete reversal of their pattern. In fact, you get a very strong positive feedback in the North American data. And so I would suggest that negative density dependence is not a pervasive mechanism driving diversity. And in fact, the normal pattern is a positive correlation between seedling abundance and trees. Now let's bring that back around to mycorrhizae. The neat thing with their data is that it's about 50-50 between ectos and arbuscular. So you can recode the data based on mycorrhizal status. When you do that, it gets a nice blur of colors there. But you do actually see that ectomycorrhizal species, as you might predict, have a slightly more positive feedback than arbuscular mycorrhizal species. But perhaps more interesting, if you look at just the data that falls below 0, remember that the data is almost perfectly split 50-50 in mycorrhizal status, you actually find that those negative feedbacks are disproportionately common in arbuscular mycorrhizal species. And so perhaps I've been doing it wrong before, that it wasn't really about positive feedback of ectomycorrhizal species, but that the interesting pattern is the negative feedback of arbuscular mycorrhizal species. And so, since we're in India, I think we'll actually have a reincarnation of the hypothesis, at least the level of being a question for the future. Let's look then at the interactions of mycorrhizal fungi and rock nutrients. And this goes back again to the late 90s with work from a number of different research groups that showed these tunnels in mineral grains that were about the same size and shape as fungal hyphae. And those were always found under ectomycorrhizal vegetation, which led authors to conclude some pretty important ecosystem effects, such as driving that rock-eating ectomycorrhizal fungi drive podzolization, that they explain ectomycorrhizal dominance, and that they can do things like alleviate acid rain effects. The only problem with that is that other authors have argued that. They've actually also shown tunnels in rocks, quote, from locations where there have been no roots, bacteria, nor fungi for the last 10 million years, leading that author to conclude that there are no rock-eating fungi or bacteria. And actually to conclude that with an exclamation point. It's very rare that you see an exclamation point in a scientific paper. And so we wanted to test that, not so much the actual mechanism of the tunnels, but what we wanted to test was whether the use of rock nutrient sources was different between arbuscular mycorrhizal and ectomycorrhizal systems. And we had a fantastic natural experiment in New Zealand where we've got podocarp forest, which is arbuscular mycorrhizal, and nothofagus forest, which is ectomycorrhizal, and they occur in different regions along the west coast. There's a large region with no ectomycorrhizal species whatsoever. And so we could look at paired plots along that boundary between the arbuscular mycorrhizal and ectomycorrhizal system and see if we saw any differences. And I should say right off that this is actually work by Nina Kola, who's a postdoc in my lab, who unfortunately couldn't be here. What she did is she took these ecosystems, she paired them on the same parent material, the same climate, and then she looked at various indicators of phosphorus cycling. When she went into the soil, she very quickly found tunnels underneath the ectomycorrhizal tree species, which was great. And then she found tunnels underneath the arbuscular mycorrhizal ecosystem, which was unexpected. So we see tunnels even in the absence of ectomycorrhizal fungi. But what we're really interested in is differences in nutrient uptake. The rock that we're interested in is this one. This is mineral apatite. It's the primary source of phosphorus in all ecosystems. And so what we did is we did a substrate bag experiment. We put sand into bags, amended it with apatite of two different types, either high or low solubility, and put that in a bag that only hyphae could enter, and then put it in an outer bag so we could capture the roots around it. We incubated those in the forest soil for a total of 12 months. We went back and recovered them, dug them up, and brought them back to the lab. First thing we did was we looked at the fungal community, and I have to say I hate doing hyphal length measurements. We got some of the messiest data I've ever seen. There was a very slight difference in hyphal lengths between the two mycorrhizal statuses, but it was not even significant. We've also done molecular analysis. The 454 data is in progress, but we've done the TRFLPs on this. We see very nice separation of the fungal community between the ecto and the arbuscular mycorrhizal system, as you would expect, but no evidence of a differentiation by substrate. And that ties quite nicely into some work out of Vollander's group by Berner that just came out in Mycorrhiza, where they showed that apatite-amended bags incubated for 5 years, so a much longer experiment, showed increased hyphal fungal colonization when you put apatite, but no effect on the fungal community. So that if there is rock dissolution going on by these fungi, it's a very widespread trait. Nothing seems to be specializing on it. What about uptake into the roots themselves? So we had this outer bag where we could recover the roots that were pressed around our substrate bag, and we were looking specifically at rare earth elements. This is the line on the bottom of the periodic table. Because those are taken up along with the phosphorus, you can actually use them to trace phosphorus uptake from the apatite. And you can look at the ratio of different elements and to trace different substrates. We looked at the lanthanium-erbium ratio, the slope between those 2 points. Here's what the results look like. We got a very nice differentiation with low solubility apatite. So there's a substrate up at the top. And if you look at the boxes, what you see is that there's clear evidence of the plant lanthanum-erbium ratio deviating towards the added substrate, showing that there was plant uptake from the apatite, but that deviation was equal between ectomycorrhizal and arbuscular mycorrhizal systems. We also looked at the europium anomaly, which is this little deviation in the rare earth elements. And again, we saw a deviation of the plants towards the substrate, but no difference between the two mycorrhizal types. So in total, we find tunnels under both ectomycorrhizal and arbuscular mycorrhizal vegetation. We find no effect of apatite on fungal community composition, and we have evidence now of equal uptake of apatite by both arbuscular mycorrhizal and ectomycorrhizal roots. So this doesn't mean that rock-eating fungi aren't important, but it does mean that they're not a good explanation for differences between ectomycorrhizal and arbuscular mycorrhizal ecosystems. So to that extent, I have to reject the hypothesis. I'm not quite willing to go as far as put an exclamation point. Okay, so it's looking pretty grim right now. We've got 1 question mark and 2 X's. So let's hope the organic nutrient cycling works out a little bit better. This, like so much else, goes back to work by David Reed, where he showed that ectomycorrhizal fungi could access organic nutrient sources. And there's been a lot of work on this by a whole number of different people. In fact, the first experiment I ever ran was looking at protein used by ectomycorrhizal fungi, and it's great fun. If you use a cloudy protein source, you can actually see fungi clearing this out of the growth media. But Petri dishes only go so far. There's a nice paper out recently by Talbot et al., where they've actually looked at this in the soil, and what they've shown is that enzymes involved in protein use were positively correlated with the richness of ectomycorrhizal species, but not correlated with the abundance of saprotrophic species. So that's great. I think probably most people in this room believe that organic nutrient use by ectomycorrhizal fungi is important. And yet, as I mentioned earlier, it's not included in ecosystem models. I actually went out and searched. I went through every ecosystem model of carbon and nutrient cycling that I could find, and I found exactly 1 that included organic nutrient use by ectomycorrhizal fungi. That was a model called KenW, developed by Mikko Kirschbaum. And what he put in a small flux of soil organic matter directly to the plants. He actually told me later he did it just to get the model to work because it wasn't balancing. But anyway, after I found his model, I also found out that he had just moved to Landcare in New Zealand, so he was actually my colleague. And so we developed a collaboration along with Kate Orwin at Lancaster University. And what we did is develop this model called MyScan, mycorrhizal status, carbon, and nutrients. And the important point in all this spaghetti of lines going everywhere is that we put in a flux from litter and organic matter to mycorrhizal fungi. And then Kate's been testing this at different levels of soil fertility and at different types of litter, either low or high quality litter. And what she does is she measures the change in the parameters when you turn on organic nutrient use. So in this case, she's looking at plant carbon and soil carbon. She saw that when you allowed plants access to organic nutrients via mycorrhizae, it caused a significant increase in plant carbon and a significant increase in soil carbon, regardless of fertility or litter quality. And that was largely due, particularly the soil carbon was largely due to an increase in total carbon inputs, which is a pretty boring result. It just shows that because we were adding more nutrients, we got more growth of the plants, and that led to more carbon input. But what was exciting was when she looked at the nitrogen to carbon ratio of both the litter and the soil organic matter. And what she showed was that by allowing ectomycorrhizal fungi to mine these resources for nutrients, she was actually causing the residual organic matter in the soil to decline in the nitrogen to carbon ratio. All of those results were consistent across all levels of fertility and litter quality, but when she actually looked at the effect on saprotrophic decomposition, it was no longer consistent. And this is particularly cool, because what it shows is that the ability of ectomycorrhizal fungi to use organic nutrients is actually suppressing decomposition by saprotrophs under some conditions, but not others. And so it gives us the ability to start predicting when ecosystems might change and when they might not, and how they might change. It may also explain the dominance of ectomycorrhizal trees on particularly low quality substrates. So the models are fine, but do they really apply to the real world? And there's precious little data to test this model right now. However, there's this nice study out by Krista McGuire, where she was working in the rainforest. She took litter from the monodominant ectomycorrhizal forest and the arbuscular mycorrhizal mixed forest. She placed it under each canopy type, and she found very little effect of the litter itself, but very strong effects of where it was placed. So it decomposed much slower when it was in the presence of ectomycorrhizal fungi. And we've shown the same thing looking at grassland to forest transitions that we see when you take grass litter and put it in the forest versus the grassland, it decomposes much slower in the forest, and equally tree litter does the same thing, although we do find a litter effect as well. And so I would suggest that perhaps the best bet for explaining what's going to happen during ecosystem transitions is by looking at organic nutrient cycling. So just to conclude, it's been over 20 years since David Reed proposed his map of the world based on mycorrhizal types. And I think we've come an enormous way in understanding the ecology and the functioning of both ectomycorrhizal and arbuscular mycorrhizal systems. But I think the challenge for us is that we have very little understanding of ecosystems in the process of change and ecosystems where the two types co-occur in spatially heterogeneous mixtures. And perhaps that's an area where we should put a bit of a priority for research. And I would suggest particularly at looking at organic nutrient cycling. Perhaps by doing so, if we can show strong effects of these two mycorrhizal types, we can actually achieve better integration of mycorrhizal ecology into a global understanding of ecosystems. Thank you very much.
Thank you very much. I'm sure there may be a lot of questions, and we have time for questions too. So,
and I can't actually see anything because of the glare on my glasses. So.
Over here, Ian.
Okay.
I really enjoyed that. That was great. Got us thinking. So, in your distance from the trees, did you consider density, or have you considered density of the trees? I think there's a huge difference between a savanna-like forest and a Douglas fir forest or your Nothofagus forest, where the trees are a meter or 2 meters away from each other. And what we're finding in the Douglas fir forest is that the roots are going 30 meters away from a bowl. And really, does a bowl matter? And that's what we need to start thinking about.
Yeah, no, I agree with you completely. The system I looked at there was actually a forest edge. So I'm using it as a surrogate for density in the sense that as you move closer to the edge, when you're also moving closer to additional trees. Does that make sense? So if you're right up against the edge, you're near a whole bunch of trees. If you're further away, you can see it as sort of a closing triangle. But I actually think in the boreal forest that mycorrhizal inoculum is not limiting. That this sort of effect is an effect that you're only going to see at low densities of trees. So I agree with you on that completely.
All right, Ian?
Yes,
Right here. I'm just wondering how you deal with trees that are dual mycorrhizal.
This is where I'm going to
Or systems, or even systems that may be dual mycorrhizal.
Yeah, I've gotten a somewhat different view of that than most people. So if I haven't offended you yet, now I will. I actually think that there are very few plants that are ectomycorrhizal that also don't have arbuscular mycorrhizae. And I don't see it as being two boxes. I think plants evolve ectomycorrhizae. Whether or not they lose the ability to form arbuscular mycorrhizae, I don't think has been well shown. So I treat anything that's the ability to form ectomycorrhizae as ectomycorrhizal. Just as the way, otherwise you end up with this gradient from species that often form both, like eucalyptus, to species that sometimes form a little bit of arbuscular mycorrhizae. I thought it was better to just treat it just as plus ectomycorrhizae or minus ectomycorrhizae without considering the arbuscular mycorrhizae in that. Okay?
I found it fascinating that you've observed a switching over evolutionary time and historical time in the forests from ecto to AM-dominated forest types. And I'm just curious what you might think are triggers or thresholds that generate these shifts over time.
In the specific case of the podocarp to nothofagus transition, it's probably one of the most controversial issues in New Zealand ecology right now, is trying to understand why does it shift and how. There is a glacial component there in terms of the nothofagus has been slow to spread following glaciation, very slow to spread. But if I was going to say why do systems shift over, I think in my mind what happens is you start going ectomycorrhizal, you've got a few ectomycorrhizal trees, they start accessing the soil organic matter, taking up the nutrients but leaving the carbon behind, making it less available to other plants. And so it forms this positive feedback cycle where they continue to drive the residual litter to more and more, to less and less of availability for other plants. Does that make sense? It almost fits in our star theory.
Thanks, Ian. It's Andrew Smith over here.
Great.
You've covered, a really excellent talk, and you've covered a lot of ground. Now, a lot of the research is done in temperate areas where the soils are really quite young, thanks to glaciation. Now, thinking back to the tropics, where the soils are much older, and as you pointed out quite correctly, you can have very big biodiversity. Now, how do feedback effects fit in, where, for example, in Southeast Asia, you have largely AM forests with some ditrocarps as well? Do you think there's a sort of climatic element following climatic change in the past, of course?
Yeah, very much so. At this point, I'm wildly speculating. If you look at the west coast of New Zealand, that large area where we've got just arbuscular mycorrhizal vegetation, that's temperate, but what characterizes that area is a lack of seasonality. And I suspect that that's very important, that seasonality creates a period where decomposition is slowed down because of the decomposer organisms are too cold, and that those cold periods might really favor ectomycorrhizal species. So I think that ties in as part of the mixture. We're actually currently looking at European data, looking at the transitions through the Plio-Pleistocene boundary and the loss of arbuscular mycorrhizal species over that time, and it does seem to correlate with seasonality. But that's very preliminary, just speculation.
I'm here.
Okay.
So thank you for this very nice lecture. My question would be that I think you confirmed and showed very nicely that availability or degradability of organic matter seems to be a key function for explaining distribution, occurrence of ectomycorrhiza and so on. But if you see this property as it is distributed in ecosystem, are we addressing the right scale? Because this property is varying a lot along soil profiles, but even within one horizon, at micro scale levels, you may have a patchy distribution of small ecosystems, which are extremely different. So my question would be, to trigger this kind of question, are we really addressing the right scale? Should we perhaps think to make more soil fractionation and trying to describe more properly microhabitats and soil and their occurrence, their balance?
Yeah, it's a tricky question. And I think fundamental to it is whether you believe fungi are microorganisms or not. I tend to believe they're not. That, in fact, the whole thing about being a fungus is that you're large and that you bridge between different resources and you move things between resources. And that may be very much an ectomycorrhizal view, but that's what makes fungi interesting, is that they're not microorganisms, that they're actually big, that they move carbon from plants out into the soil. They can colonize one patch, and they don't view that as an ecosystem, but as part of a much larger ecosystem. So I'm actually not sure that the micro scale is the right scale to work on this. But the challenge for me is trying to take some, to work at the ecosystem scale, to work at the larger scale, and see if things that we see at small scale scale up. And I accept that other people are more interested in working at the small scale, and that's incredibly valuable because we can't do the ecosystem ecology without also understanding the smaller processes as well. Okay.
Some other Some other question? Yes, over there.
talk. Great talk, Ian. You're getting such a good workout up there, I couldn't help jumping in to participate, grilling you.
Someday I'll learn to speak slower so that I have no time left for questions, but I always speak too fast.
So in thinking about the different roles of AM versus ectomycorrhizal fungi versus saprotrophs in decomposition, would you care to take the leap and draw any speculative conclusions about importance for carbon sequestration?
Of ectomycorrhizae?
Well, yeah. I mean, let's take a high latitude system and say, do ectomycorrhizae overall contribute to increased carbon storage in soil? If you somehow tweak the system to go towards more saprotroph dominance, would you get more or less storage?
I actually took that slide out because when I practiced the talk, it took the full time and then some. We actually have some data on that. The pattern's the opposite of what you would predict. And it's strange. Everybody expects that when you get ectomycorrhizal forests, you'd get more soil carbon. But if you're actually looking at young ecosystems, that is those that haven't had a long time to establish, you get a loss of soil carbon, not a gain of soil carbon. That's really well established from pine plantations, that there's about a 20% loss of soil carbon. It's been unclear to me in the past if that was actually due to the ecosystem shift or due to something about the planting of the pine trees, the disturbance. But we're now looking at invasive pines, and we see exactly the same thing, a very rapid loss of 20% of soil carbon. And what's interesting in that is that they're actually mining out nutrients. We're looking at phosphorus fractionation. We're actually seeing movements out of the most recalcitrant pools and into available pools. So they're using, it indicates that they're using carbon from some pools that we thought were completely unavailable. What happens over the longer term could be quite different. And that may be as much as anything driven by the accumulation of fungal hyphae and necromass. But I don't have data on that. I think there'll be a few talks later on today that are going to address that. So.
I don't see any other questions, so
okay.
it's a good test for you. Thank you very much.